Good evening, viewer. Welcome to our Glory Globe TV. Here's the breaking news. Yes, scientists at the Hospital for Sick Children are delving deep into the non-coding genome to unravel the complex genetics that underlie blood pressure regulation and hypertension. Yes, the leading cause of cardiovascular disease affecting 1.25 billion people worldwide. While the human genome comprises coding and non-coding DNA, the latter has long been overshadowed by its protein coding counterpart and is commonly dismissed as junk DNA. In recent years, scientists have identified several genomic regions that are associated with high blood pressure, including differences in DNA, also known as variants in the non-coding genome. A research team led at Sick Kids by Dr. Philippe Maas, a scientist in the Genetics and Genome Biology program, set out to understand how these variants likely regulate blood pressure genes and in doing so describe regulatory processes in the genome for blood pressure gene regulation. Their findings, recently published in the journal Cell Genomics, offer a compelling glimpse into how these genetic sequences influence the regulation of genes associated with high blood pressure. This research unveils for the first time the intricate connection between how variants in the non-coding genome affects genes that are associated with blood pressure and with hypertension, explained Maas. What we have created is a kind of functional map of the regulators of blood pressure genes that cannot only inform future investigations into cardiovascular genomics, but that also presents a framework that can be applied to study other genetic conditions. Despite consulting 98% of our genetic material, the non-coding genome does not actively produce proteins. Instead, non-coding sequences regulate coding genes in various ways. While more traditional genome-wide association studies are used to identify associations between genetic traits and specific diseases, they are unable to describe how genetic variants act and influence nearby genes. Findings from GWAS have informed much research on the function of variants in the coding genome, but very few have expanded to include variants on non-coding regions of genome. Maas and his team used massively parallel reporter assay technology at Sick Kids and computational expertise from Dr. Mastra Malay at the Barcelona Supercomputing Center to examine genetic variants in the non-coding genome at a massive scale and identify how they likely regulate blood pressure genes. By leveraging stem cell expertise from Dr. James Ailes, a senior scientist in the Developmental and Stem Cell Biology program, the team was able to study the genetic variants in human relevant heart cells. The study, which reviewed more than 4,600 genetic variants with the high thought-out technology, represents one of the largest endeavors to date in investigating and assigning functions to the non-coding genome. Surprisingly, Maas notes the analysis revealed high densities of variants located at genes tied to blood pressure regulation. With the growing adoption of whole genome sequencing, we can find thousands of new variants in a single genome, says co-author Dr. Seema Mittal, staff cardiologist, senior scientist in the Genetics and Genome Biology program, and scientific lead, Ted Rogers Center for Heart Research and head of cardiovascular research at Sick Kids. Discerning which variants may be linked to disease holds the potential to enhance the utility of genome sequencing in clinical settings. BNS report, Glory Globe TV, Delhi, India.